Okay, good morning. Kia ora, everyone. We've got someone else joining us too. Welcome to our first ERI seminar for this year. We're really lucky to have Dr. Kevin Wakeman visiting us here at the University in Tauranga. Kevin's from Waikato University and he's been coming to New Zealand for 8 years. I think you said on a summer field trip. And so we've just been hosting him here this week. And he has agreed to give us a talk about his research, which is all about alveolates, which is something I must admit I don't know that much about. So I'm looking forward to learning a bit more about alveolates. And I think from what I understand, it's just ecology and biology of alveolates. Kevin's been at Hokkaido University in Japan for about 9 years now, working on this. So yeah, lots of interesting research to talk about. I'll pass it over to Kevin, and then following Kevin, he's also got one of his PhD students here, Sean Cunningham, and Sean's also going to be doing a bit of a talk about his research too. So we'll have 2 parts. We'll do Kevin's talk first and then followed by Sean. So I will hand you over to Kevin, and we can just make this smaller if you want, so it's out of the way.
Perfect, yeah.
Or even just minimize it. Oh, there we go.
That'll work. Perfect. Yeah. So thank you very much. Thank you everybody for coming. Because it's such even a very cozy group, feel free to ask questions at all. So I have the microphones and everything turned on. So don't feel shy about asking a question if I go by something a little quick or you'd like to know a little more about a topic. So there's really no problem at all. So yeah, so thank you again for coming. I was going to talk a little bit about the biodiversity and evolution of marine alveolates. And this has a lot to do with some of the work that I've been doing up in Hokkaido. Originally, I'm from the US. I've been living in Hokkaido for the last 9 years. Before that, I was at University of Tokyo for a year, and then at OIS, so the Okinawa Institute of Science and Technology. So I think collectively, I've been in Japan for almost 12 years, 12, 13, yeah. It's been a really great time. And right now I'm working in the Biodiversity Division in Sapporo. And so off to the side, those are the professors that are in the group. So Takeo and Kazuhiro and Abe Sensei also. And so we work on various aspects of biodiversity. We're focused on microalgae, macroalgae, and I do a little bit of that and some other protist groups, the apicomplexans. So this is a bit of the current group. So Aika is actually my wife. We met in Canada. So she works in the lab as a postdoc and researcher, and Eric, Sean's here with me. P. Srati from Thailand, Yusuke, and Kehin and Talia. I'm going to be going a little bit through their work as well, and I'll try to give them credit when we're talking. And then later, Sean's going to be going over some of his research. So I kind of got started in the marine environment. I guess sorry for the noise. I didn't expect it. I kind of got started in this doing some scuba diving work when I was an undergrad. And I really enjoyed working with invertebrates. And I wanted to actually go through graduate school and work somehow with invertebrates in some aspect. And I had this opportunity to study organisms that live together with invertebrates. So this idea of symbiosis. And the group that I specialized on as a student were the apicomplexans. Apicomplexans are notorious for things like malaria. And we know from the mosquitoes and human interactions and also apicomplexans in livestock, but they're also very prevalent in. And so that had an interest. And I thought that it'd be really fun to couple my kind of love for the marine environment and an interest also with these parasites and make it relevant into a PhD project. And so this kind of led me down this pathway of symbiosis, so understanding how organisms live together, and this was from an aspect of invertebrates, but also from microeukaryotes, so like apicomplexans. And so when we're thinking about symbiosis, we're really talking about this close and long-term interaction between organisms, and there's these mutual symbioses. There's also commensal symbiosis and parasitic, and they differ with regard to what each individual in that symbiosis is getting out of it. So mutualists are interactions where both partners are benefiting or multiple organisms are benefiting. Commensal is something where you may not have a direct benefit or harm but they're still the organism or a part of the organism is being used. And so one popular example is hermit crabs that can use the shell of moss, and also parasites. So parasitic interactions can be where one organism is having their fitness lowered by another. And so my work on apicomplexans was looking at these parasitic or obligate parasites within the marine environment. So this is where I worked at in Canada. And so we did a lot of cold water diving, which was actually my one of the reasons actually I moved to Okinawa, is I was so excited to get out of the cold water. I just wanted to be in warm water for once in my life. And I've actually never really gone back to the cold water. I don't think I kind of got away from that. And so to introduce the alveolates, this traditional view of alveolates is that, or very general view rather, is that it comprises three main groups. So these apicomplexan parasites, these dinoflagellates that have free-living, heterotrophic, and also parasitic lineages, and ciliates. And then when we're looking at apicomplexans, they're incredibly diverse. So it's not just the malaria or plasmodium parasites that we know about with regard to humans. Those are prevalent to us or important to us because, of course, the human health risks. But every organism or every metazoan, these are multicellular animals, plays host to at least one apicomplexan parasite. And a bit of a thought exercise is that when we look at humans, we find that there's 4 or 5 different apicomplexan parasites that specialize on us. So things like Plasmodium, Babesia, Toxoplasma, also Cryptosporidium and Coccidia can all parasitize humans. And so if we were to extrapolate those numbers and spread them over all metazoans that we know, we can really quickly see that there are hundreds of thousands at least species of apicomplexan parasites, and these are going to have different levels of interactions, and of course be important even in fisheries. So from a public health perspective and also economically as well. So yeah, they're found in different types of like sea turtles, also marine mammals as well. And kind of the question that I wanted to get at in my PhD or beginning this research was understanding where this diversity came from. So how do we go from, when we're looking at alveolates, how do we actually make transitions from something that is inherently free-living or mutualistic? How do we go from that to something that is an obligate parasite, such as the Plasmodium that infects humans? And what are these changes that have to occur at these different points? And I thought studying marine apicomplexans was such a really interesting point or nexus point to study, because it represents one of the earliest stages of obligate apicomplexans. And so when we're looking at, I guess if you're looking at this transition from free-living photosynthetic to parasites, the marine apicomplexans are one of the first lineages that have a necessity to actually be a parasite. So they don't have a free-living capability. There are other lineages or close relatives, sister groups to the alveolates, that are free-living. And so we know that these evolutionary transitions had to have occurred over time. And I wanted to study one of the first. And so those are the groups that invaded the invertebrates. So I thought that was kind of an interesting nexus point. And also looking at how you actually move through the hosts. So how do you go from something that is in coral to something that might infect an arthropod to something that infects a vertebrate? And while that may seem easy, there's a lot of inherent challenges to that. So things like the early metazoans don't have a true gut. So how do you infect organisms that have different feeding strategies? Arthropods have various life cycles and stages and larvae. So how do you transition between those? And how do you go into something like a vertebrate? Many vertebrates, for example, can lead terrestrial lifestyles. And so you have to modify the way that you're living together with an organism, but also be very plastic enough to fit the lifestyle changes, but also make transitions into new environments. And so these evolutionary transitions from alveolates, in particular the apicomplexans, are kind of interesting because they're changing drastically with regard to environment and host, and they're able to do this very successfully. And so this is an example. This is a Chromera avilia. This is one of the earliest sister groups to apicomplexans. So if we're going to go from alveolate, or if we're going to go from apicomplexans and look at the nearest sister group that's still photosynthetic, this is Chromera. And interestingly enough, this was held in culture because it's not an obligate parasite. They're photosynthetic. You can look at the brown pigment on the cells. This was actually thought to be a symbiodinium culture for several years. It was just held in somebody's culture collection. And when they did the 18S sequencing, they realized that it actually branched close to a sister to apicomplexans and not within dinoflagellates. And that's where that symbiodinium or the zooxanthellae would branch. And so this was actually a really interesting organism at the time because it represented a really good evolutionary transition. So how do you go from kind of a missing link, that photosynthetic ancestor, to this obligate parasite? So a lot of work was done on Chromera, and then they found also other sister lineages too, so Vitriella. And so how these plastids are changing in the evolutionary history. So this really gave a lot of insight into those molecular changes that need to occur. One of the questions that I always end up getting about apicomplexans is asking me, well, if they're so diverse, are they all really that harmful? And that's a really good question. And when we're writing grant proposals, the short answer is always yes. They're really harmful. But this is actually a bit of a nuanced answer because we don't really know. When we're thinking about pathology and the way organisms associate, when we're looking at, well, I guess defining parasites, we would define it as lowering the fitness of an organism. But if an organism is able to associate and finish their life cycle, even with the parasite present, it's almost debatable how harmful a parasite could be. Looking even at ourselves, there's examples of toxoplasma that can associate with people, but unless in only very special situations, like in a pregnant female, would that be harmful, for example, to the fetus and the baby? And so I don't know if you're familiar, if you go to the doctor and you're pregnant, oftentimes the doctor will tell you to stay away from cats or cat litter, and that's because of toxoplasma. But inherently, toxoplasma is not harmful unto itself. Only in very special situations. And so when we're looking at apicomplexan parasites, they may or may not, or they would have different degrees of pathology present within it. And I'm going to give I'm going to try to give an example of this. So here's the life cycle of the malaria. And within invertebrates, if we were to take a cross section through, this is a Nereis worm. If we look in the gut, central part here, I highlighted it with arrows, you can see these apicomplexan parasites, and they attach. They still have You have the bigger? Yeah. Oh, no, it's okay. So they have a modified apical complex at the anterior of the cell. This is a mucron, and so they're attaching to this gut lumen, and they can also find their way into the coelomic spaces of these invertebrates. Here's a good example of a terrestrial apicomplexan in a dragonfly, and they form cyst stages, and then the more elongated ones, these are the feeding or trophozoite stages. These are harmful in a way is that they change the ability of the dragonfly to conduct sugar metabolism. In effect, it causes something analogous to diabetes within certain arthropods. And so when we're looking at mollusks or different invertebrate groups, such as the arthropods, mollusks, some of these apicomplexans can be quite benign, while others can actually cause different types of abnormalities or pathologies where they're infecting. So if you're an arthropod and have an infection in your reproductive organs, these can affect your ability to produce gametes or the survivability. So there can be varying levels of I guess, harmfulness to those hosts. One really good example and something we've been working on in Hokkaido is scallop. And there was this is a study from, gosh, I guess 2015, so about 10 years ago, where they found a die-off of scallops. And when they did the pathology or necropsies on these dying scallop, they found a large number of apicomplexan parasites. And this is a really typical situation where, at least in marine environment, and some of you might work on parasites, oftentimes they don't really look for the pathology unless there's a problem. And so when you go in and look for it, there's sometimes a bit of affirmation bias, I think, where they find a sick organism, they look at it, and then they find apicomplexans. And apicomplexans can be really common. And if you have a lowered immune system, so you might be, if you have a lowered immune system, those apicomplexan parasites can be really harmful as well. And so sometimes we have a situation where correlation is not causation. And a normal immune system can handle a certain apicomplexan load to a large degree. But if you have a compromised immune system, so HIV, that might make toxoplasma more harmful to you as well. And this might be the same, or this is the same with different scallop populations. And so this initial paper was just questioning the idea of are apicomplexans responsible for these die-offs of these Icelandic scallops. And it was a really interesting idea, but the consensus was that it was correlated with it. So the presence of the apicomplexan, yes, was correlated with a sick and dying scallop, but they weren't really sure if that was the actual cause of the die-off. And so later on, this was a follow-up to a very similar study. This was done, I think, just last year by Espinoza, and they did a lot more detailed study where they were able to experimentally look at the infection rates of the apicomplexan within this is on the East Coast of the United States. And what they found is that it did actually correlate with the help of the scallop. However, they also found other things. They found that these infection rates or the pathology also correlated with the stress on the organism or heat stress. It also correlated with hybridization effects. So how diverse are your populations? If your population has a high degree of genetic variability, it would be able to handle, for example, an apicomplexan infection. If you have some types of genetic bottleneck where you have lowered amounts of genetic diversity, that might more greatly impact the apicomplexan infection. So the answer to this is that, yeah, apicomplexans can be really bad. However, it's a bit more nuanced, and these might be more related to, or in some cases, more related to things like stress and the ability for the immune system, and also hybridization, and your population might play a role in that. And so this actually was something that we were thinking about when I started working with New Zealand. So Steve Webb and then Jonathan Banks, and this is a song. And they contacted me, I think in 2015, 2016. They go on record, I think Song, at least, is one of the only people to read my PhD thesis. And that made me feel really good about myself. And so she contacted me. She did her PhD on shellfish pathology on green lip mussels here in New Zealand. So she was at Auckland University, but stationed down in Cawthron Institute in Nelson with Steve and John. And we wrote a grant to look at apicomplexan infections between New Zealand and Japan. Some of the impetus of that was trying to compare source populations. So when we're looking at shellfish, a lot of shellfish were moved around the world with the shellfish trade. And we wanted to kind of take a look at, are these populations from Japan? Are they also found in Japan? And where are these located? Are they just in New Zealand? The green lip mussel, I believe, is endemic to New Zealand. And so this was a really good opportunity to take a look at endemicity and how parasites, how specific are they to their different hosts. And so Song was able to come over to Japan and look at some of the shellfish populations where we worked in Hokkaido. This is apicomplexan X. This is some work that Steve was actually doing on green lip mussels down at Kaufman Institute. And as you guys are probably aware, green lip mussels are really important economically and also culturally in New Zealand. And so there's a really good reason to study these, at least from a economic perspective. And when we're looking at why that's important, or the apicomplexan pathology, these did correlate with lower ability, or the fitness, or lowered fitness rather, of the green lip mussel. So it did lower the ability for the green lip mussel to produce gametes. It's also impossible to eradicate. So it seemed like whenever these infections were found, they were never able to be rid of. And so this is, I think it was the MPI, is that the organization? So whenever they would find these infections, whole lines of oysters and green lip mussels were being taken out. And so it was having a pretty big impact on the fisheries in that area. Additionally, there was also additive effects with Bonamia. And so Bonamia is another pathogen. And whenever apicomplexans and Bonamia were found together, the infection was greater than the sum of its parts. And so there would be these 5 or 10 times more pathological effects of the organisms whenever these parasites would kind of gang up in these host populations. And so Song, this is actually, I guess, yeah, kind of a mock one of version 1 rather, of the Wakeman lab. And Song, and they came over, we did some work. John couldn't keep a straight face during our photo ops. And we also had a great opportunity to come down to the South Island, New Zealand. I think it's Cable Bay. And we were specifically looking to try to give a genetic identity to apicomplexan X. And so one of the main points of the project, and what Song's PhD was really based on, was trying to find out, is apicomplexan X an apicomplexan to begin with, because its morphology is actually a little weird and bizarre for what it is. And what she did was she took PCR, or she found by cloning out PCR products an apicomplexan sequence. And then what she did was she hybridized the probe to the tissue. And so what you're looking at over there on the right-hand side of the slide is just in situ hybridization of this apicomplexan sequence that was retrieved by just cloning PCR products. And we were able to take that, or Song was able to take that and hybridize it to these apicomplexan infections. And so the blue there are clusters of apicomplexan cells that are labeled with this methylated adapter on this probe. And so the way that it's prepared, it looks a little funny with the histology, but that's because it's been dehydrated. But so the blue in that photo are those apicomplexan cells, and that gave a molecular identity to that organism, which was kind of really interesting. And then some of the work we're trying to do later was identifying what are the life stages. And this is something that, gosh, it's still kind of ongoing. We're trying to figure out where and how these apicomplexan are getting into the green lip mussels. And apicomplexans can be a little confusing. Some of them have very complex life cycles. So they have a direct host and an indirect host, like malaria, and also coccidians, which is where these go. But they can also have just one host. And we don't really know at the moment if this APX and the green lip mussels have 1 or 2 hosts. And so it's one of those things of absence of evidence is really hard to show, because if we can't find this APX in a secondary organism, it would make us believe that, okay, well, maybe it's just one organism. But we don't really know that to be sure, because if we screen these invertebrate communities for the APX genetics, but we don't find it, we don't know if it's a false negative, if we just haven't picked the right invertebrate yet. And so right now, I think where it stood is that we're just trying to look at different worms. And I think one of the candidates is going to be looking at something like a mollusk, like a snail. And that was the case with the Icelandic scallop, as it was actually going. So probably the next step would be looking at those. And yeah, and then so we expanded this project a little bit to look at scallop. This was some work done out in 2021 by students in my laboratory looking at a Keshi scallop. We looked at, I guess, the scallop just in the bay there. We were able to get, I think we had over 100, 139 different scallop. And we wanted to look and try to marry the more traditional approaches to more contemporary molecular work. A lot of pathology is done with histology, and that is really great for looking at if it has some sort of effect on the host. One of the issues with that is it's really difficult to give a molecular identity to things. And we don't know if these are just different life stages or different parasites of the same thing. One thing we tried to do was look at laser dissection microscopy. So we can take a laser, or we can take a histological slide and use lasers to cut out in very high detail certain parts of that histological section. And it uses kind of this modified, uses a kind of a modified lid to an Eppendorf tube, and you can push, like cut the section out and then push this lid down and pick it up. And you can see where those sections match the cutout region, and then you can extract DNA from it. Yeah, it's a really neat technique. One of the shortfalls of it, though, is that a lot of the former or past histology was done with formalin, which makes it really hard to get DNA from. However, there's a modified technique using methanol, so alcohol fixations. And that was really good for both fixing the histology and getting the DNA out. And so there's it's called a methylcarn, but it's essentially alcohol-based fixation of the tissue. And so if anybody is doing that in the future, I'd highly recommend using that methylated stuff. And so looking at parasites, one of the questions I wanted to look at also was how these organisms speciate. One thing that I was looking at in my PhD was just how many parasites are there. And I was kind of always wondering, why is it that there aren't just one parasite to go across everything? Even when we're looking at Plasmodium, there's several species of Plasmodium parasites that specialize on different organisms, birds, mammals. And so I wanted to take a look at how exactly these radiate and jump into different groups, and if there's any type of pattern. So the photo that's up there, these are the different, or some of the different apicomplexan parasites that I found in my PhD. And there is actually a little bit of a pattern with how they look like. There's the ones on the bottom that are worm-like, and then there's essentially everything else. And I wanted to see if it's actually predictable when you go to a host and look for these. Are you able to find certain patterns in their evolution? And the short answer, I won't make this point too laborious, but the short answer is yeah. It is really highly predictable that when you go to the ocean and find a terebellid worm, or you go to terrestrial environments and find a beetle, or pick up a tunicate or an echinoderm, there's a large degree of host specificity within these organisms. And so it appears that apicomplexans kind of jump and radiate through different host groups. And this pattern is actually very host specific, down to the level of species. And even organisms that overlap in range can have a different parasite associated with it. So they're very picky, it seems, with the host that they associate with. And even in hosts, and this is on a level of species that have speciated relatively recently, the parasites seem to follow them as well. So they're very loyal to their hosts. And so when we were looking at the different parameters I was looking at, or different options I was looking at, is the different types of speciation you can have is you can co-speciate. So that means that when your host speciates, the parasite will speciate with it. You can also speciate by jumping over and switching hosts in that. And you can also speciate within the same host. And so those are kind of the different options that a parasite has. And within the same host, for example, you can just specialize on a different organ. So within some worms, we even see that they're able to go from the gut, and some of them specialize just in the crop. So they go to the anterior part. So there's a type of niche partitioning that these organisms can have. I wanted to, and this is actually part of Eric's PhD, what he's going to be working on, is answering a question about or looking at different degrees of host specificity in coevolution or co-speciation. And one model we were looking at was tunicates to look at this. And one of the reasons we were looking at that was because there's a very high degree of host specificity from some of the preliminary work that we had. There's also a high infection rate, and they're cosmopolitan. And that benefit simply means that we're going to have a very high number of examples. And so it gives us a lot more statistical confidence when we're doing analyses. And the other one is tunicates can't run away. So one of the ways we want to set up the experiment is that we increase the number and the ease of doing this. So we don't want to chase around fish, for example, in the ocean, if we can help it. Some of the preliminary work this was based on was work we did in Canada. And when we grabbed different tunicates, even in a very similar region, Canada, we found that the parasites were, in fact, very loyal to each other. And now we've expanded that. And a lot of Eric's work is going to be looking at these samples that are in the Atlantic Ocean, also the East Pacific, as well as across Japan. And even he had some samples from down in New Zealand from some years ago. And so the idea would be that he's going to be looking at cophylogenies, so looking at congruence and the pattern of if these match or don't. We would expect to see if a phylogeny matched or has a high degree of congruence, that there is co-speciation occurring within these populations. If there's host switching, which is on the right-hand side, we would expect to see some type of where the pattern or where they cross each other. And that would suggest that maybe they've radiated into tunicates, but once they're within tunicates, they would tend to kind of jump across from different species. And so these are just some of the organisms he was looking at. And then that's going to be a large part of his degree in the analysis. And this is just some of the biogeography that's going on there, as well as some of the work that we were doing in the West Pacific. Look at that. So yeah, there's still a lot left for to discover within alveolates. There are groups, for example, that have different types of what appears to be plaster organelles. And so when we're looking at how photosynthesis has changed in this group, much like dinoflagellates that can kind of disregard and take up things in their life history, I think dinoflagellates can have different types of primary and secondary and tertiary symbiote in their kleptoplasts. It might be also that different alveolates have done something very similar to that just in the distant past, but they've kept relics of that. And so one of the things that we're doing is trying to find these very bizarre lineages and look at the genomics and try to find and look at their evolutionary history and find out how they, on their path, have kind of become these obligate parasites. And so they've undergone a type of almost parallel evolution where they've done something very similar to other groups, but they've done so independently. And this is actually up in Nelson. This is a really bizarre apicomplexan, this is a very bizarre alveola. It's not an apicomplexan. And you can see the relic flagella that it has on the interior. So this organism hasn't gone through, does not have a modified apical complex. It still has maintained its biflagellate ancestry. And so the genomics of these are really interesting to us because these are these organisms at the nexus point that are going to tell us what changes have occurred in these lineages. This is a really highly modified apicomplexan parasite. It's gone completely away from feeding with any type of apparatus and it just went with kind of a flat pancake look. And it's that ball in the center, that's a very large nucleus. So one of the defining features of these gray-green parasites. And what it's done is gone to a surface area to volume ratio. So it just lives in the gut, pulsating and absorbing nutrients from the hosts. And then, of course, for other types of alveolate parasites, there's those found in squid. If you look at squid that are found offshore in their gills, this is a Hochbergia, more closely related to dinoflagellates that also has a parasitic lifestyle. This is what it looks like when it's up close. Those triangle shapes, those are the alveolar. So that's kind of the namesake of alveolates right there. So you can see these, the alveoli, there's 3 different layers to these alveolates, and then they modify it in form. I think it's like a structural thing that they're doing. And then this is a copepod and a blastodinium. So this is a dinoflagellate parasite. And what it does is it essentially turns a copepod into a greenhouse. And you have this big sausage-like dinoflagellate that infects the gut, and it takes up essentially the whole gut. And this is a death sentence for copepods. And what it's going to do eventually is just explode. And then it's going to turn these little spores that are in the bottom and then put that into the water column to be infected by, or to infect rather, another copepod. And then this is a Parkinson's, so another type of dinoflagellate that infects the abalone. And so all of these are really important. And when we take a look at the transitions that these groups have made, this is a look at just the plastids rather. They've actually all maintained types of non-photosynthetic plastids. Within the case of apicomplexans, this has to do with, I think, their sexual reproduction. So the way that they're running, it's kind of like you're cycling, you're taking different cables. I kind of imagine it like a old operating system on a telephone. And it would have made the connection a long time ago with photosynthesis. However, now they've simply taken the cables and rerouted it, and they use it for more like an asexual reproduction. And so there's still mechanisms that can be used for these old organelles, or relic organelles rather. And these alveolate parasites in general have converged upon a lot of these parasitic forms, but in different ways. And so it's kind of neat just how plastic that they can be in their population. So that's a lot of the work that I did and do. And then, of course, in the laboratory, I was going to talk a little bit about the students and then hand it over to Sean. This is work that Talia is doing in the laboratory. She's actually working on labyrinthula in mangroves. And these labyrinthula are these detritivores. They also have parasitic lineages. So when you go out and look at the seagrass wasting disease, so those brown splotches on the seagrass is also caused by labyrinthula. She's also interested in looking at biotechnology. So these produce a lot of fatty acids that can be very useful for applications in the food industry. So this DHA, that's a really popular food supplement. And so she did a lot of work in Okinawa looking at labyrinthula populations. And we found actually a very high degree of genetic diversity. So it's not just one labyrinthula that specializes in this area. And this is actually just some preliminary work. She has, I think, over 70 different sequences now representing 6 or 7 lineages. So it turned out to be a really exciting project. And then she's growing these in culture and then also going to do the chemical profiling to look at that diversity. So P. Sarati, so he's just going to start his PhD. He was going to join us in New Zealand, but couldn't make it. And he's working on symbionts that have more of a mutualistic relationship. So this is an acyl worm, and the green and brown dots, those are microalgae. The green is a tetrasomus, and the brown is a dinoflagellate. And these live together and appear to be obligate mutualists for the worm. So it's very difficult for the worm to live and complete their life cycle without these different algae living inside of it. And so it's kind of a neat project that he's working on. These worms, these acyls, they live interstitially, so between grains of sand. So when we go around, we just kind of take the top layer of sand and look for the diversity of worms. This is a video. These are, I guess, the more cute organisms in the lab. But they look kind of, yeah, I guess like gummy bears in a way. Yeah, so some of his work is really exciting. We're able to also take these cultures from the acyl and grow them. So while these are obligate for the acyl, all of these lineages of algae that live within the acyl are facultatively free living. So they can live just fine by themselves in culture. And so we're trying to leverage that so we can identify them, but also use them for different types of chemical analysis. And P is originally from Thailand. And so we've been trying to do a little bit of this work in Thailand to get a little bit more of an idea of the biogeography and try to find out a little bit about the species diversity, as well as what type of effect biogeography might play on this. some of his work has found that there's actually a whole lot more diversity. It's not just one tetrasomus that lives together with those, but also I guess several different lineages. And the same is true for the dinoflagellates. So on the right-hand side is the Symbiodinium. These are the same organisms that associate with coral. And so very analogous to coral, it's likely that these Symbiodinium play a role in providing photosynthase for their acells hosts. And then he's also going through and trying to identify these organisms that are very, very challenging to identify morphologically and even genetically. And so that's really, that's what I had to talk about today. I was going to very quickly just thank the people in Canada and also different students that have helped out with the projects. Of course, people in New Zealand that really started this whole work. And also people in Japan and Okinawa. And we have really great opportunities to conduct research with boats that take us to, or boats that enable us to get samples from the deep sea, and also sample really rare or hard to access areas. And so I was going to talk a little bit about Hokkaido, and then I'll hand it over to Sean. But Hokkaido is in central Sapporo, so it's the largest university. This is a caveat by area in Japan. It's also a really nice area to do research. So it has really pristine coastline, farming area. For activities, I guess it's got Sapporo beer and the different beer gardens, and also the winter and skiing. So it really does have 4 seasons that make it a really appealing place to be. I guess that's one of the reasons I've hung out for so long there. They also have a lot of really neat resources. So Shoro Marine Laboratory is a great place to conduct field work, and Sean will talk a little bit about that. And also the Akechi Marine Lab, which is more the flagship marine station that we have. And then, yeah, this is the snow festival that just happened actually, I think last week in Sapporo. Just for scale, those black dots in there are people. So they build these really gigantic snow sculptures. And yeah, that's about all I really had to talk about. If you guys have any type of interest in going to Japan, especially for PhD or postdocs, I'd really highly encourage that in general. And so, yeah, that's what I was going to do. And then I guess if anybody has questions, then we'll get Sean set up. And if anybody has any questions, I'll be glad to answer them while Sean's going to switch.
Thanks so much, Kevin. That was really fascinating. Yeah, I'll just quickly switch over our talks so we can do Sean's one. Oh, there we go. Oh, sorry, I'll just switch off that screen. Sorry. Just share Sean's presentation with you. Cool. Hopefully you guys can all see that one now. So I'll just turn you around and introduce Sean Cunningham, who is Kiwi, but is now living and studying in Japan at Waikato with Kevin. So yeah, take it away, Sean.
Yeah, I'm Sean Cunningham. I'm studying harmful benthic marine algae in Japan. I'm originally from Auckland. So I'm originally from Auckland. I done my bachelor's degree and my master's degree at the University of Otago. After that, I done some work in the rock lobster industry for a couple of years, and then I went to Environment Southland, and I was like, worked a lot on Undaria Peninsula, the eradication program down there, and some stuff around just around like marine biosecurity, biofouling management, and also some terrestrial biodiversity projects. Then through that work, I linked up with the Clawthorne Institute and got a role in the marine biosecurity team. We done like vessel biofouling management for working with MPI, and then some work with the aquaculture industry and that kind of thing. Then yeah, after a few years there, I was just looking for a change, and there was already a established relationship between Waikato University and Cawthron through Kevin and John and things, and there was an opportunity to do a PhD overseas, which I had some interest in doing, and so I applied for some funding there through the Japanese government. You can the mixed PhD scholarship, which is funded by the Japanese government and available to international students pretty much all around the world. But yeah, for my research, I'm interested in harmful marine algae, in particular dinoflagellates, and that's these guys in the top right of this slide. Many of them a certain number of them can be harmful and form things like red tides and use up oxygen and or produce toxins and that kind of thing. But in more recent years, we're seeing these blooms occurring in higher latitudes with warming waters and that kind of stuff. And so in Hokkaido, I think in 2021, there was a Karenia dinoflagellate bloom that resulted in like large shellfish die-offs and urchins and that kind of thing. But that's a planktonic species, and then my project's focusing a bit more on the benthic side of things. So there's species like or groups like Gambia discus and Ostreopsis and Coolia. They're sort of found on I guess macroalgae, corals and that kind of thing. I think you may have heard about like ciguatera fish poisoning, and that's these groups are largely associated with that. Ostreopsis is more like can get in like respiratory infections in people, but also has been associated with like urchin die-offs, and it's found can be in quite high numbers in New Zealand as well. And there's been some work done on that with the Auckland Marine Lab. I think next year's done some work. But yeah, the bottom two pictures here is like a benthic dinoflagellate bloom. And so yeah, I guess this slide's just like a kind of an overview of what I'm doing and where I'm sort of going. A large part of it's going to be focused around Hokkaido. And last Northern Hemisphere summer, we done a bunch of sampling all around the coast of Hokkaido, looking for these toxic benthic dinoflagellates. My focus is largely on those 5 groups, like Ostreopsis, Procentrum, Amphidinium, Coolia, as these are known to be harmful. And then the methods are more sort of down the bottom, so we collect them and isolate them and culture them, and then we can do some genetics work, know what they are, and look at the morphology, so their plate, particularly these, at least 4 of those species, armored dinoflagellates, so that you can look at their plates and try and distinguish differences in their morphology. But the genetics is also really helps with that. And then once we get an idea of what they are and if they're interesting, then we start to bulk culture them, use methanol to take out the bioactive compounds, and do like toxicity assays with various organisms. And so over in Japan, we do a lot with brine shrimp just to get like a crude, if it's toxic or not toxic kind of approach. And then we're doing some work with Cawthron with the aquaculture group there, looking more at like oyster settlement or early development, that kind of stuff. And then I guess the last bit where we're looking to this summer is to look at some of the bloom dynamics of Ostreopsis in Hokkaido, as it's quite a very seasonal place where you get really cold winters, really snowy, but these toxic species are still persisting in the environment. And so yeah, this is just sort of where we've sampled to date and what we've found. So Waikato, lower diversity, mostly Ostreopsis, Amphidinium, and Parasitrum, and then down in Okinawa in the subtropical areas, we're getting a huge variety of like Gambia discus and Ostreopsis, multiple species of each clade, heterocapsule, so all sorts of stuff living down there, but still toxic in both, and potentially more recent studies are showing that some of these species are pushing more polewards, I guess, and not a lot of work has been done particularly around Hokkaido, but also in Japan itself. So we're just adding a bit more information there. One of the main projects I've been working on since I've been here is looking at Kulia in Okinawa. We basically sampled around the islands and isolated a bunch of different Kulia, and then done the genetics and microscope work, and we identified 5 different species from that region, 4 already described, but a couple that are new to or not described in Japan, and then one that hasn't been described before, which is pretty cool. And so, but yeah, these Kulia are really widely dispersed around the world. I think about the only pattern that really exists is that is sort of this Kulia monotus seems to only really exist in the Mediterranean and the Atlantic, and then Kulia maliensis seems to be quite really broadly distributed across tropics and higher latitudes. So even down in New Zealand, we've got Kulia maliensis all the way down Stewart Island, but it's also really abundant in the tropics. And then there's a few other ones that seem to be more focused around the equator and things like that. And some of the work, the work that we've done here, we looked at the morphology quite in detail, and at least within the Kulia monotus clade, where the novel species was found. It was the morphology was really similar, and the plate patterns and the sizes really quite similar between maliensis and palmarensis and our novel species from the Shigaki. And so, but the genetics kind of shows a bit of a different story, so might be a bit hard to see, but the undescribed species fits in between Kulia palmarensis and Kulia centrocrocea monotus. We've done a bunch of toxicity bioassays on this stuff, and very variable or very minor toxicity in these upper ones, but then Kulia maliensis are very, very toxic to at least brine shrimp, and that's just some of the bioassay data. So we expose brine shrimp to the extracts, the algae extracts, and this sort of mortality rate over time. So done the 72-hour exposure, but quite clearly Kulia maliensis on the right here is showing quite a toxic effect, and then the rest of our ones not so toxic, but some toxicity shown, or a moderate level of toxicity shown, at least to brine shrimp, but they are quite hardy. And then we've also done some work on the Hokkaido Ostreopsis as well, so looking at their toxicity, just to get a broad overview of what's toxic, what isn't. And these are just strains from the various sites around Hokkaido, and we've done some just different concentrations. So yeah, but all of them were toxic to brine shrimp at least, and then we want to move on to doing some assays with. Well, we have been moving on to some assays with the Cawthron Institute. So I think last December I done some work in New Zealand with Anne Pugnier in the aquaculture group, and we exposed the male gametes of oysters to various extracts. So from New Zealand and Japan, so we had a Kulia from Apicomplexans and Kulia from Japan and Okinawa, and some Ostreopsis from around New Zealand and some Ostreopsis from around Japan, and we exposed the male gametes to these extracts over like a 1-hour exposure. It's quite a different assay to that brine shrimp, but we saw like a really minor effect, and we're largely thinking that it's the sperm have like chemical defenses and things like that don't allow it to be such a huge effect on them. And then the next step we want to do, look at larval settlement and things, to see species of benthic and how that's interacting. And then from here, this summer we're going to look at Ostreopsis bloom dynamics in West Waikato, in Offshore Bay, and the idea is just to count, I guess, to get this sort of algal bloom chronology when it peaks, when it stops, when the bloom starts and stops in Hokkaido. We're going to look at using these like artificial substrates. And so it's just like a fiberglass screen, and some previous studies have shown that like Gambia discus, these cells over like a 24 or 48-hour period, they will colonize the substrate, and then you can remove it and capture it, wash the cells off, and yeah, and then do a cell count and see what number. And then the idea is to deploy that over sort of April to November-ish, and then do cell counts and get an idea, and then genetics, toxicity, things, and we'll be focusing on things like Osteopsis, parasitic, and whatever else really shown up in the bay. But it was one of the areas we sampled last summer, which gave us the idea. And so yeah, so just to wrap up my main, I guess my projects that sort of, at least the data's kind of wrapped up as the sort of diversity of Coolea and diversity of Osteopsis in Okinawa, and then some of the this initial look into the toxic benthic dinoflagellates in Waikato, and then we're moving on to more of this I guess impact of the benthic dinoflagellates on shellfish, and also their sort of ecology in the Waikato region. Yeah, but that's my talk, and yeah, any questions I can answer.
Great, thank you so much, Sean. We're kind of right towards the very end of our time slot, so I'm just going to there again. So if we've got any sort of burning questions, we've probably got time for one or two. I don't know if anyone here has any questions. No? Okay, otherwise I might wrap it up and say thank you so much to both Kevin and the right way, and Sean for coming and visiting us, and for sharing their a little bit about their work on apicomplexans. I never knew there was so much diversity and so many interesting things to do with them, so it's been really an interesting talk for me. And yeah, I guess if anyone's got any questions, you guys can contact them in advance by email, sorry. But otherwise, thank you guys for all coming along to the seminar today. It's been really nice to have you here, so thank you, and yeah, we'll see you next time.
Right.
Oh, sorry, Georgia, you've got a question? Oh no, just a clapping. Great. Great, thanks very much, guys. We'll see you later. Thanks, Maria. Stop the recording.